So today we are going to look at the practical session number 8, which is blood and bone marrow. So the objectives are as follows: describe the components of blood as well as the function of each of them. Number 2, discuss the stages of development of cellular elements and give the description of each stage. Number 3, describe the structure of erythrocytes and conditions affecting them. Number 4, classify leukocytes and describe the function of each. Number 5, describe the light microscopic myeloid tissue. Okay, myeloid, we're talking about bone marrow. Okay, so those are objectives for today. All right, so in this practical session, we're going to look at two sources of us where we can study blood and their components. So we have a source of blood from the peripheral. So it's known as a blood smear, peripheral blood smear. So this is blood that is outside the bone marrow. Then also have a bone marrow smear. This is collection of a sample of blood from the bone marrow itself. So we all know that in the bone marrow, this is where all blood cells are formed. Then in blood vessels, that's where now mature blood cells are circulating. So we're going to have a source of blood from the vessels that is called peripheral smear. Then we're going to have a source of blood from the bone marrow, known as the bone marrow smear. That being said, this is a preparation of blood from those two samples that I had talked about. So let's look at the blood smear. So there are various stains that are used to stain cells, and most of the times, components of the cells, the nucleus, the cytoplasm, and their organelles will stain either basophilically, that is, they stain densely, okay, or they will stain acidophilically or eosinophilically. So basophilically, it will be purplish or blue, whereas eosinophil or acidophilic, it will be, red or orange. Okay, so those are the two stains that we are going to look at. So the first one is a blood smear. Remember, the blood smear is a blood sample gotten from the blood vessels. So it's known as a peripheral smear. It is not gotten from the bone marrow. So what are we expected to see here? Okay, so in this sample, we find that, so this is a peripheral smear. So peripheral smear, blood has been spread out on a slide, then it is stained. So we're going to see various types of cell. So like in this example here, what we are seeing is a blood smear. So we see these, almost of equal size. These are red blood cells. So the blood smear will be filled mostly with red blood cells. So in the central portion, majority of the cells are red blood cells. Now, since we have also white blood cells, so white blood cells are large, so usually they are pushed to the periphery as you are smearing the blood because they are large anyway. Okay, so these are red blood cells, or otherwise known as red corpuscles. The white blood cells are different types. So like this one is a white blood cell. So white blood cells are known as white corpuscles. Then we have smaller ones, these minute cells here. So these are platelets. They're usually in clusters. They appear together. They like clumping together. So these are platelets. So now let's begin to break down blood one by one. So let's begin with red blood cells. So this is a blood smear, like I was saying, peripheral smear. Here we are now focusing on the red blood cells. So this is a high power magnification. So the first thing you should do is identify red blood cells. So saying there's a majority. Okay, so red blood cells here are easy to identify. Okay, so here we're saying they are pink stained and they lack a nucleus. If you observe, there is no nucleus here, there's no nucleus there, no nucleus in the others as well. So we see that the central region is pale, whereas the peripheral region is more densely stained or thicker. This is because of the shape of the red blood cells. Red blood cells are said to be biconcave disc-shaped cells. So the middle portion is thin, whereas the peripheral portion is thicker. So if you stain the cell, the peripheral portions will pick up much of the stain because they are thicker, whereas in the middle, it is thin and therefore to be less stained. So that's why they are appearing like this pale central region with a densely stained peripheral region. Now, because the relative size of the red blood cells remain the same, the red blood cells are used to measure. So they're used as a standard of reference to measure the size of these other cells of blood, okay, and not whether they have some problem, some pathology or not. So these are used as standard, cells. Actually, even when you're talking about isotonic, hypertonic, and hypotonic solutions, you consider the concentration of solutes inside the red blood cells. So you need to draw at least, 3 or 4 cells. So you can draw this one, 1, 2, 3, 4. Make sure that they are drawn like that and label them as such. So these are white red blood cells, with some islands of platelets. That's a platelet, that's a platelet, that's a platelet. But at this stage, only draw the red blood cells. Let's look at the neutrophils. So at high power, this is still high power, trying to isolate the neutrophils. Now, neutrophils are part of the white corpuscles, the white blood cells. So neutrophils are large. And we're saying these are the majority of white blood cells. So majority of the white corpuscles are neutrophils. And these cells are said to be multilobulated. The nucleus has multiple lobes. So some books will say polymorph nucleus because they have multiple nucleus. So like you can see here, it is multi-lobed. This one lobe, another one, another one. So 3 lobes like that. So that's why it's called a polymorph nuclear cell. Then apart from that, these cells contain small granules in their cytoplasm, okay? In this case, the granules of a neutrophil stain basophilic. You can see that they are bluish or purple in color, okay? Then we're saying if you examine this cell, and it's well stained, you can even identify a sex chromatin known as a bar body, okay? So a bar body is a condensed X, chromosome that can be seen. So some books will say a drumstick. In the manuals, you find say drumstick, sex chromatin, or bar body. So a bar body is a separate or detached part of the X chromosome. So in here, I can't see it, but we can see that there are lobes, 3 lobes, 1, 2, 3. So this is a neutrophil. So draw at least 1 or 2. Now, in this case, we're only seeing 1. So it will suffice to draw 1, or just draw it 3 times if you like. Okay. But in the online link that we gave you, you may identify other neutrophils in that, and you can draw them. Maybe they may have a different shaped nucleus, but at the end of the day, these are neutrophils. What else are you supposed to look for in a blood smear? We're supposed to identify eosinophiles. So eosinophiles, the name eosino is gotten from the type of stain that it picks up. So this picks up, instead of picking up a basic dye, it picks up an acidic dye. So it stains eosinophilically. So majority of the cytoplasm and their organelles will stain with the acidic dye or acidic stain. And that's why we are seeing that the color is different here. But look at the size and compare to the red blood cells. Red blood cells are smaller as compared to the white blood cells. So these are eosinophils, okay? And they are bilobed. You can see there's this lobe and another lobe. But this whole thing is a nucleus. Then we have granules inside the cytoplasm. These granules are staining eosinophilically as compared to the neutrophil. So we need to draw this one as well. Next, identify a basophil. Now, basophil are very difficult to identify in common stain. Now, this was focused direct on a basophil, but if you're given a microscope, sometimes it becomes a bit difficult to isolate a basophil. Nevertheless, a basophil is also, some books will say bilobed, but the nucleus is irregular. So it seems as if it is a bilobed nucleus. The other thing is that the nucleus is very difficult to identify because the cytoplasm is filled with basophilic granules, and these granules occlude. They almost blur the nucleus, that you can't see exactly the shape of the nucleus. So that's why it's given the name basophil, because it picks much of the basic dye. So it will stain bluish or purplish. Neutrophil, it was picking both eosinophil and basophil stain. But basophil specifically picks up the basophilic, stain, whereas the eosinophil picks up the eosinophilic stain. So this is what you need to draw. Demonstrate the nucleus, which is shadowy there. It's not so clear because of so many granules inside the cytoplasm. Then show the granules as well. So as you are drawing neutrophil, basophil, and eosinophil, make sure that you also draw the surrounding red blood cells for comparison's sake. So all red blood cells near these cells must be drawn. Next type of cell we're looking at is a monocyte. So we are now seeing that a monocyte has a kidney-shaped nucleus. So the nucleus is distinct. It's not occluded by any granules. So monocyte has no granules in cytoplasm. So the nucleus becomes clear. As we can see here, the cytoplasm is stained basophilically like that, but the nucleus is very clear. So books will say kidney-shaped or bean-shaped nucleus. So the nucleus is not cut in half to give us 2 lobes or 3 lobes. It's only as an indentation here, then it has this shape. So this is a monocyte. So make sure that as you draw these 2 monocytes, draw the surrounding red blood cells as well. Okay. Even some islands of platelets, like this one and that one, and this one should be drawn as well. Next is we look at the lymphocyte. Now, saying lymphocytes are small. So lymphocyte in terms of size, lymphocytes are closer in terms of size to the red blood cells. Okay. Apart from that, we're saying lymphocytes are a bit larger than red blood cells, and they stain darker than red blood cells. Red blood cells are pinkish, but these will be purple and bluish in terms of stain. So they have a round nucleus that almost covers the entire cytoplasm. So this is a lymphocyte. You see that only small portion of the cytoplasm is left. So they have a large nucleus and only a small part of cytoplasm is left. Okay, so you need to draw at least 3 or 4 of these and the surrounding red blood cells. So these are red blood cells, and then this is a lymphocyte. Next cell we're looking at is a platelet. Now, platelets are small clotting cells. Okay, so they are very small. These ones, they are actually smaller than red blood cells. Okay, so they appear round or oval. Okay, and they are scattered during a blood smear preparation. So as blood is being smeared on the slide, you find that the platelets are spread evenly throughout the smear because they are very small as compared to the white blood cells. White blood cells, they are usually pushed to the periphery, whereas red blood cells fill up the whole space because they are the majority of cells in blood. Okay. Apart from that, we're saying that platelets have no nucleus. So they are non-nucleated cells, similar to red blood cells, except that when you stain a platelet, it's going to show 2 distinct areas. Number one is that it has a central basophilic zone. So you see that the central portion here stains basophilically, bluish or purple, whereas the peripheral zone stains eosinophilically. Okay, therefore they are given 2 different names. So the central zone is known as the chromomere or the granulomere because it's said to have some granules, and those granules pick up the basophilic stain, whereas the surrounding cytoplasm stains with acid. So it is given as hyalomere. So we have granulomere and hyalomere. So granulomere stains basophilically, hyalomere stains eosinophilically. So we need to demonstrate this here is highly magnified. So this is a platelet, one platelet that was isolated. So make sure that the central zone is shown as granulomere, whereas the surrounding one is hyalomere. Central zone, basophilic. Peripheral zone, eosinophilic. So you need to draw at least 2, 3, 4 cells. So you can draw this one, that one, and this one. And make sure that you also draw the surrounding red blood cells. So we have finished the part of, blood smear. Now let's look at a bone marrow smear. So we are now taking a sample of blood from the bone marrow, where we have a chance of seeing cells that are not yet mature, because these cells found in the peripheral, in the blood vessels, these are mature cells. Okay, so we call it a peripheral blood smear. Most of the cells you find there are mature, but the bone marrow smear, you're going to find also immature cells.